Welcome to Chapter 2. Chapter 2 is divided into 4 sections. In this video, we're going to focus on the first one, the properties of light. Visible light spans a spectrum of colors. Like think of a rainbow. We've got red, orange, yellow, green, etc. Understanding the properties of light and subsequently being able to manipulate light has really allowed us to be able to see really small things or things that are really far away. We also have additional techniques such as staining that will allow us to enhance microbes or parts of microbes that we're interested in seeing. And this has really allowed us to produce really high detailed images that are important to science and medicine. We'll be looking at many different types of microscopy in this chapter. The one you've probably encountered in your previous classes is bright field microscopy, where cells or the specimen is stained and the background is not stained. A new one that we're going to talk about later is dark field microscopy, where the specimen appears bright against a dark background. By the end of this section, we should be able to describe EMR, electromagnetic radiation, and also describe how lenses are used to manipulate light. We also have another clinical focus problem in Chapter 2. This time we have a 17-year-old camp counselor, and she is playing basketball, hurt her knee, and it looks like it's getting more painful and swollen over time. She goes to the nurse. They see a lot of pus coming out from the surface, so maybe it's infected. So the nurse looks like she swabs the wound to get a sample. They're going to send it out for lab testing, right, medical lab, and they're probably going to analyze it under a microscope. So think about what we might be able to learn by these bacteria by looking at them under the microscope. What do you think? Maybe, I don't know, what do you think? The size, maybe the shape of the cells? We're going to learn more throughout the chapter. Light is really interesting because it can behave as both a particle and a wave. And if you take physics in the future, you'll learn more about this. Visible light is made up of electromagnetic waves that behave like other types of waves. So there are several important properties of waves, including wavelength. So the wavelength is the distance between one peak to the next peak. So that is one wavelength. And wavelength is often abbreviated with the Greek letter lambda. So let me draw it again. It looks like this. Another important property is the amplitude. The amplitude is the height of a wave peak. And I can see from here to here, so from basically down here to up here is the amplitude of the wave. You can also measure the trough, so the depth of the trough. That is the amplitude of the wave. Frequency is another important characteristic, and it tells us the number of wavelengths or wave cycles per unit of time. So if this is some arbitrary unit of time, we have about, let's see, from here to here is one wavelength, another wavelength here, about 3 wavelengths in that first wave. And down here you can see there are many more wavelengths per unit time, so that's a greater frequency. Frequency is related to energy when we talk about light. The higher the frequency, the greater the energy. And I'm going to abbreviate energy NRG, because when you say that, it sounds like energy, right? So greater energy when you have higher frequency. Lower frequency, what do you think? The lower the frequency, the lower the energy. So high frequency, high energy wavelengths would be things like maybe UV light or X-rays. So very strong, very high energy, things that you wouldn't want to get exposed to too often. Whereas low frequency waves would be things like microwaves, or what else? Radio waves. Those are some examples. Light waves can interact with things by being reflected, absorbed, or transmitted through the material. Reflection is shown in this middle picture, and you can see light is bouncing off the surface of the object, such as the example our book gives us, where if you're wearing a red t-shirt, for example, it appears red to our eyes because the red is being reflected off of the shirt. Absorption is when the material captures that light energy, and you can see that here. You can see light being shined onto the sample, but nothing passes through the other side because the light has been absorbed. And our book also gives an example where if you've ever played with glow-in-the-dark toys, These plastics absorb light and they actually re-emit the light later on. And then transmission is shown on the right down here. You can see transmission occurs when light can pass through something that is clear or transparent. It also depends on the thickness of the object as well. For a fun fact here, it's often interesting to students to learn that green plants actually don't use green light for photosynthesis. The reason these plants appear green is because they reflect and transmit the green wavelengths of visible light, of that visible light spectrum, because these plants actually primarily use red and blue light for photosynthesis. Here we can see a petri dish that's transparent, so light can pass through the clear petri dish and we can see through to the other side. So that is transparency. For opacity, we can see this opaque meteorite, and light cannot pass through the material, so you cannot see the hand on the other side of the meteorite. Light waves can also interfere with each other, and when they do that, they form these interesting patterns that kind of look like ripples in water when you drop a rock in. So one of the most common experiments is shown here, called the two-slit experiment, and I can see two openings here and here that light can pass through. When that happens, the light waves will often interact with each other because they'll be out of phase with one another. So imagine, let me change colors, imagine this one produces light waves that look like this, and then the other opening produces light waves that either look like this, or they could be in sync, right? When that happens, we have interference, and our book doesn't get into the details, but there's two basic types. So imagine when the white light waves are in sync, the green and the blue that I kind of drew here. If they're in sync, then we get the result of constructive interference, and that will lead to a greater intensity in the light, which usually appears as brighter light. But if they're completely or perfectly out of sync, then you can see the result is this straight red line in the middle, which is usually perceived as darkness. And that leads to these interesting bands or patterns of light that we see down here. The diffraction of light can also happen when light is passing through a small opening, such as these shown here, or if it's bending around a small object. And diffraction is more pronounced when the object's smaller or when the opening is smaller than the wavelength, because if the object or the opening were big, then the wave could mostly go straight past through it. But if it's small compared to the wavelength, like these shown here, then you can see there will be a lot more bending through it or spreading out around it. And ultimately, the diffraction will produce a region similar to what we saw before, containing dark and bright bands of light. All right, let's try this checkpoint. If a light wave has a long wavelength, let's draw two, that one versus this one. Remember, oh, that doesn't look great, but the wavelength is from the peak to the next peak, so that's one. Here's another wavelength from here to there. If it has a long wavelength, is it going to have a low or high frequency? Right, frequency is per unit time, right? So if I choose this as my arbitrary unit of time, about this length, right? I have two wavelengths per unit of time here. This is time, but I have one wavelength per unit of time here. So it's going to have a lower frequency, right? Lower frequency. And then we said low frequency is associated with lower energy. Okay, so low energy, low frequency for longer wavelengths. Down here, if an object is transparent, if it's transparent like glass or plastic, light is going to go through it, right? Which one is that? Reflect, absorb, or transmit? That is transmittance. So refraction is very important to microscopy, and refraction is the bending of light. And this usually happens when light is passing through objects of, for example, different densities, as it's going from air to water, or maybe air and then through glass. And the bending or the refraction happens because there's a change in the speed of the light waves passing through through the different medium. All right, so how much the light bends depends on many things. It can depend on the angle of the incoming light. So if the light is incoming this way, that'll affect the bending as it passes through the next medium, or maybe if it's passing through this way, the bending will change as well. And how much light is bent depends on the refractive index of the object. And this tells us how much the light speed will slow down as it passes through that object. Generally, the refractive index is dependent on density. It can depend on the wavelengths of light that are being passed through. It can depend on temperature. And the greater the difference in the refractive index between two types of medium or media, like air versus water or air versus glass, the greater the bending will occur. We can see refraction here on the left where we've put an object, let's say it's glass, on a table, and you can see light will bend as it passes through the object that has a different refractive index. On the right, I can see the incident ray of light, so this is the light coming in. It is refracted, it's bent, as it passes through an object of a different density, right? A different medium. And light is also reflected off of this object. And just for fun, what's always true is the incident ray angle is always equal to the angle of the reflected ray. Our book also gives us an example of this pole, and it's supposed to be a straight pole, but it appears to be bending through the water because of that difference in the refractive index of air versus water. And you might have seen this at home, either maybe at the swimming pool or if you're drinking water through a straw, right? It appears to bend as the light passes through the water. So it turns out that when light enters a material with a greater refractive index, it's going to bend toward the normal. And the normal is an imaginary line that's perpendicular to the interface, to the border between the first surface and the second surface. And I can see this medium right here must have a greater refractive index because as light passes through it, it's bending closer, right, toward that normal line. We're going to see that in microscopy, lenses used curved surfaces in order to allow light rays to meet or converge at a single point, and that's going to be our focus. So focal point, we're going to see more on the next slide, are where the parallel light rays are going to meet after bending. The focal length, I'll also show you in the next slide, but that's going to be the distance from that lens to the focal point. And then I'll show you a variety of lenses in the next slide as well. Lenses are like prisms, and they take advantage of the fact that light bends as it passes through a different medium, the medium with a different refractive index. We have two types of lenses shown here, convex and concave. Convex lenses are thicker in the center and thinner at the edges. In contrast, concave lenses are thinner in the center and thicker at the ends, right, at the edges. Convex lenses, how they work is they can bring parallel light rays and allow them to converge, to meet at our focal point. Concave lenses do the opposite, and they actually cause light rays to diverge, to spread apart. In anatomy, we learned that the human eye has a lens that's able to focus light onto the retina in the back of the eye. Microscopes are similar, but they're more powerful, right? We take advantage of the convex lenses in microscopes to bring light from very close objects into focus, right? We can't do that with our eye. If you put something right in front of your eye, it'll be blurry. But microscopes can focus on these very close objects and allow them to appear larger to us. And that's how magnifying glasses work too. There are a lot of other details that are beyond the scope of our class, so don't worry about calculations or things like virtual images that you might have learned about in physics. What I would like you to know are there are two basic types of microscopes that you might have encountered. There's something called a simple microscope, which just uses one convex lens. In our classes, we use compound microscopes, and compound microscopes are microscopes that have two or more convex lenses. So the first convex lens is usually like the eyepiece or oculars, right? That one could be the ocular or the eyepieces. And the second one would be those objective lenses. And we usually have 4 on our microscopes. The 4 times, 10, 40, and 100 times objective lens. And these allow us to achieve much higher levels of magnification so that we can see details in our specimens. The image focus and the size of the image depends on the distance between the object and the lens, as well as the curvature of the lens. If you bring an object closer to the lens, the focal point will be further from the lens. And then the greater the curvature of the lens, the more curved a lens is, the shorter the focal length. And this will make the image look bigger, so larger images will be produced. So remember that the focal distance or focal length is the distance from the lens to the focal point. I like this picture from Leica because it kind of shows us everything all together. We have our slide or object here, which is first focused by the objective lens. The objective lens actually produces an image that is picked up by the second lens, the eyepiece or ocular lens, and that is going to be focused by our human lens so that we can see an image. It's picked up by the retina. And ultimately what we see is a magnified image of the original object. And you may have learned in previous classes that everything we see under the microscope is upside down and inverted. So I think one of the most common ways that we do this in lab is you look at a slide with the letter E on it, and under the microscope it appears upside down and inverted, left and right. Remember that visible light is just one form of electromagnetic radiation. We also have things like radio waves, microwaves, etc. And these are organized on the electromagnetic spectrum based on their wavelength and frequencies. Visible light actually just takes up a small part of that electromagnetic spectrum between the lower energy infrared waves and the higher energy UV rays. Remember that wavelength is the distance between wave peaks, so from one to the next wave peak. And frequency is how many oscillations you have per unit time. So this would be something like a high frequency wavelength, or waves, I should say, and these would be low frequency or lower frequency waves. Remember that higher frequency waves have greater energy, right? So shorter wavelengths is associated with greater energy, and this means more energetic photons. Photons are the particle form of light, right? They're particles or packets of light. And the colors of visible light correspond to specific frequencies. Red is low energy, low frequency light, whereas violet is high energy, high frequency light. When you have white light, this is actually a combination of all of the colors of the visible light spectrum. And these can be, or white light, can be separated by a prism, I'll show you in the next slide, into a rainbow. Remember that the refractive index of a medium not only depends on the density of the medium or temperature, but also the frequency of light. So here we have a prism that has a different density than air, and it's going to separate or bend the white light into its individual colors because of their different frequencies. This is known as dispersion, that bending of light through the prism to separate white light into their individual colors. Shown here is the electromagnetic spectrum organized by wavelength and frequency. And within the EMS, the spectrum, we have the visible light, right? Visible light here. And that is magnified up here. If I look at relative wavelength and frequency, I can see very small wavelengths here, which correspond to high frequency and higher energy, like gamma rays or X-rays. Whereas on the other side of the spectrum, we have greater wavelengths, so much bigger wavelengths, which correspond to lower frequency and lower energy, like radio waves. Some materials can convert non-visible electromagnetic radiation into visible light, and that happens through fluorescence or phosphorescence. Fluorescence is when UV or blue light excites the electrons of the material, and these electrons move to a higher energy state. When they come back to their ground state, energy is released, and that is released in the form of visible photons, light that we can see. These emitted photons have a lower energy and frequency compared to what was originally absorbed. And we take advantage of this in biology to visualize certain tissues or specimens or cells. So we have something called Texas red, for example, that absorbs blue and emits red light, a lower energy light compared to the blue. Or fluorescein isothiocyanate, which absorbs UV light and emits a lower energy green light. There are some materials that can go through phosphorescence. This process is when the emission of light is delayed after absorption. So glow-in-the-dark plastics work this way. So the photons are emitted following a delay after the absorption of the original light. Here we have an example of multiple fluorescent dyes being used to stain a specimen. And one example is DAPI. DAPI actually absorbs UV light and emits blue light. We also have that fluorescein isothiocyanate that absorbs UV and releases green. That takes us to our next checkpoint, which has a higher frequency. I remember that green is right next to blue, and blue has high energy, which means it has a higher frequency, so green light's going to have a greater frequency. Why does dispersion occur through a prism? Remember that each wavelength of light, each color, has a different refractive index. So as they bend through the prism, they're going to be separated. They bend differently. And then fluorescent dyes, why do they emit a different color of light than they absorb? So when they absorb light, electrons are excited, and then the electrons fall back down to their ground state, releasing a lower energy light or frequency of light. And this happens because some energy is lost during the process of exciting the electron and allowing it to fall back to its ground state. That takes us to the end of our first video for Chapter 2. In the next video, we'll finish Section 2.1 and get started on 2.2 and 2.3.